Allora, io cercherò di essere più semplice, di parlare in modo più semplice possibile, mi scuso per gli scienziati, ma lo scopo è soprattutto far capire alle famiglie la neurobiologia anche della protocaderina 19, per cui scusate, ma cercherò di essere veramente basic. Allora, nel sistema nervoso centrale, come ormai tutti sapete, i neuroni sono le cellule che
For the cells,
le cellule si chiamano neuroni e queste
and
ricevono e
the cells, the
e
neurons,
mandano degli
receive and
stimoli,
send out stimuli
comunicano tra di loro,
because they communicate
ricevono degli
one
stimoli
with another,
che li trasmettono
and
e i
they
siti di
transmit the stimuli that they have received.
sono i siti
And
di comunicazione,
and the
sono le
loci,
sinapsi.
or the
A me
sites,
piace sempre
the
far
communication
vedere
sites,
queste
are
immagini che è
synapses.
molto
I
esplicativo.
love
Questo è un
showing this
ramo
image, which is
di un
truly
neurone
indicative. This
andando a
is a
vedere
branch, a
ad alto
neuron branch,
ingrandimento
and if we
potete
magnify
vedere
the
che esistono due tipi
locus,
di
we see that
sinapsi,
we have two
sinapsi
different synapses.
excitatorie e
We have the
sinapsi
excitatory
inibitorie.
ones and the
Qual è la
inhibitory
differenza
ones.
tra le due
Now,
sinapsi?
what is the
Le
difference in between the
sinapsi
two synapses?
excitatorie
The
utilizzano
excitatory
come
synapses tend to
neurotrasmettitore
use as a
il glutamato,
neurotransmitter the
le
glutamate,
sinapsi
whilst
inibitorie
the inhibitory
utilizzano come
synapses use
neurotrasmettitore il GABA.
the GABA
Andando a
as a
vedere ad alta
transmitter.
risoluzione
And if we then
potete
have a further magnification and higher resolution, you can see what's the difference in terms of image. The excitatory synapses are much more dense, and the inhibitory ones, especially in this image, are much more regular. They have a regular pattern. They're not that much densely packed. Then at molecular level, let's look at their organization. The glutamatergic synapses, the ones that release glutamate, are much more complex, and they contain many more proteins, whilst instead the inhibitory ones are more simple in their structure.
Che cosa il corretto funzionamento nel sistema nervoso centrale?
In terms of central nervous system and its functioning and the normal cognitive function, let's see what happens there. There is a perfect balance in between excitatory synapses and inhibitory synapses. Now, what happens when we have an imbalance in between the two types of synapses? What happens is that we may have different types of defects. We may have something that goes in favor of one or the other, the inhibitory type or the excitatory types, or both synapses may even appear to be less performing, less functioning. So the final element in all three conditions is represented by a cognitive defect. And if we show the X chromosome and we look at the chromosomes and genes expressed, and especially the mutated ones, the ones associated to autism and mental retardation, what we notice is that 50% of the mutated genes code for proteins that are synaptic proteins. In my laboratory, we've been studying and characterizing from the molecular point of view a number of genes that are associated to autism and mental retardation. Now here, we have been pointing, and this is the TISPAN-7, and the other one is SHU4. We still have to publish the complete data. Then we have oligofrenin, and more recently, that is to say a year and a half ago, we started a new project on Protocadherin-19. Today, I will be referring to the data, the highly promising data, which have got to do with the molecular mechanism, having to do with the functioning of Protocadherin-19. In this slide, I just want to show you the family of Protocadherin. There are 71 elements, all of them grouped into different
e non
clusters. We have the so-called clustered protocadherins and non-clustered protocadherins. The number 19 belongs to this group, the non-clustered protocadherin. And the difference is that the clustered protocadherins have a specific genomic locus, whilst the non-clustered protocadherins that do not present with this characteristic. And then another differentiating element is that as far as the protein is concerned, if this is the structure of the protein, we see that there are a number of identical segments that belong to the extracellular part. In the clustered protocadherins, the segments are 7 in total. In the non-clustered instead, we may have more than 6. Then there is a dominion known as the transmembrane dominion, which is going across the membrane. And then there is another part, which is known as a C-terminal portion. Now, what do we know from the neurobiology of the Protocadherin-19? We know that these studies have been accomplished in the zebrafish pattern and modality. And we are here interested in looking at the expression of protocadherin at brain level. So we did use some homogenate and some layers of
Vedete che la
rat
proteina
brain.
nel cervello è
And
ben
as you
espressa
can see,
e
the protein is
soprattutto
well
è
expressed in the
molto
brain.
espressa
And
nelle
it's
due
very
regioni
well
che sono
explained, especially
corteccia e
in two
ippocampo, che
regions,
sono
i.e.
quelle
the cortex and the
deputate
hippocampus,
per
that are
i
the
processi di apprendimento.
regions where the learning and the
Dopodiché,
mnemonic processes
quindi la
take place.
protocadherina è ben espressa
We said the
e
protocadherin is well
questa
expressed
inoltre
here.
abbiamo visto
And
facendo dei
then, on top of that,
frazionamenti
we
cellulari
were making and accomplishing some cell
è
fractioning
presente nello stesso
procedures, and we've
frazione
seen that
in
in the
cui
same
sono
fraction,
presenti altre componenti
this
delle
protein is
sinapsi.
expressed together with other
Questo
components of
classico
synapses.
componente delle
And
sinapsi
this
è
is a classic
5, quindi è un altro marker
component
che è
of
presente in sinapsi.
the
Dopodiché siamo
synapses
andati a
is
vedere
the
il
PST95,
livello
which is a
della
typical
proteina
marker.
in due
Then we
condizioni,
checked
nei
for the level of
neuroni
the protein
giovani,
in different
in cultura
conditions,
e in
that is,
neuroni maturi.
cultured
Cioè,
young
il nostro
neurons
scopo è di
and
vedere
other
se l'espressione
mature
della
neurons.
proteina
So the purpose
varia
was to
o
see whether the
variasse
expression of the protein
tra
could vary or
durante
would
lo
vary
sviluppo e i neuroni
between
più
the
giovani in cultura
development
sono
in
più
young
preparati,
cultured
più
neurons that
osservati.
have been
Quindi dopo pochi
placed
4 giorni
in a
dopo
dish, and then
giorni
they
in
are
cultura,
again
oppure neuroni
observed
maturi
after
che hanno
being
delle
placed in a
sinapsi
culture,
molto
or whether they are
complete
expressed in
e funzionanti
synapses,
e
mature
mature.
synapses, where the synapses are
Andando a
complete and
vedere, non
mature.
so se potete apprezzare
Now, I
bene,
don't know
però
whether you could
il
fully
colore in
appreciate this
rosso
image,
è
but what's marked here in
lo
red
staining
is indicative of the
l'anticorpo
staining
specifico per la
of a specific
protocaderina.
antibody for
In questo potete
protocadherin.
vedere quindi che la proteina è presente
So you
sia
see
nei
that
neuroni giovani
the
che nei
protein
neuroni
is
adulti.
present in
Dopodiché,
young
nella
neurons,
seconda
but also in
fase
adult
della
neurons.
nostra
Then
caratterizzazione
after
siamo
that,
andati
we
a
had a
vedere
second
se la
stage
proteina è
where we did
espressa
try
sia
and
nei neuroni
see whether the
inibitori, quelli
protein
che
is
vi dicevo
expressed in
prima,
the
quindi sono
inhibitory
neuroni che
neurons,
rilasciano GABA,
that is to
quindi
say the
sono
ones that
delle
release the
sinapsi
GABA,
inibitori,
that is inhibitory
e nei
synapses,
neuroni
or
eccitatori,
in the
utilizzando
excitatory
come marker
neurons.
specifici dei
And we
due
were
tipi di
using the
neuroni,
specific markers
quindi
for both
GAD
types of neurons,
67, un
that
marker
is the
specifico che riconosce i neuroni
GAD67, which is a
inibitori,
specific
e
marker
un
recognizing
anticorpo contro la
neurons, and
solubilità
another
di una
antibody
solubilità
against
specifica del
the
recettore
specific
AMPA,
subunit
che
of the
è
AMPA receptor,
classico diciamo
which is
marker di una
the
sinapsi.
typical marker of an
Allora,
excitatory synapse.
in questo ingrandimento potete vedere
In
che la
this
protocadherina è
magnification, you can see
espressa
that
in entrambi i
protocadherin
tipi
is
di
expressed
neuroni,
in both
però
types of
è
neurons,
altamente
but it is
maggiore
then
nei
highly
neuroni inibitori.
expressed in the inhibitory neurons.
Allora, il nostro, quindi, detto, dimostrato in
Now,
modo banale anche che la protocadherina è espressa in tutti e due i tipi
once
di
we've seen that
neuroni,
the protocadherin
siamo
is
andati
expressed
poi
in both types
a
of
vedere
neurons, having seen this, we
andare
then
più a
wanted
studiare
to
in
check
dettaglio la
for
funzione
more
della
details. We
protocadherina
wanted
nei
to see the
neuroni.
function of protocadherin
Abbiamo
within the neurons.
utilizzato un
And
classico
we utilized a
approccio
traditional
biochimico
approach, a
che
biochemical
usiamo di
approach,
routine in
which is what
laboratorio,
we
cioè
use
il 2-hybrid
in the
screening.
lab. That is
E
the
con questo sistema
2-hybrid
è
screening.
possibile, con questo
And
test, è
with this
possibile
test, it is
trovare
possible to
degli
find
interattori possibili,
some
interattori
possible
di molecole,
interactors
possibili interattori cellulari
molecules at cell level.
delle proteine. Quindi che cosa abbiamo fatto? Abbiamo utilizzato la coda
So
interna
we
citoplasmatica
used
della
the internal
protocadherina, che è
cytoplasmic
questa,
tail of
con
protocadherin,
questa coda citoplasmatica abbiamo fatto
And
appunto
with this
il tuo
cytoplasmatic
abbiamo trovato
cell,
che
we
la
studied
protocaderina
and we
interagisce
observed
con
that
la
the
subunità
protocadherin
alfa
interacts
1
with the
del
alpha
recettore
1
GABA.
subunit of the GABA
Allora,
receptor.
esistono due tipi di recettori, ricettore
We
GABA
have
B
two
e
types of
recettore
receptors,
GABA A.
GABA B and GABA A.
Ogni subunità è composta da
And
4
each subunit is made up of 4 different
e il recettore
TM
attivo
dominions.
completo è formato
And then
da
there is an
5
active
subunità.
one,
La
the complete
caratteristica
one, which
del
is made
recettore
up of
GABA
5 subunits.
è quello
The typical
di quando è
characteristic of the
attivo
GABA
nella
receptor
trasmissione sinaptica
is
di far
whenever
passare
it
lo
is
ione
active at
cloro.
synaptic
Quindi
level,
cosa
allows
succede
for
in
the
modo molto
passing of
semplice?
the chlorine
Quindi
ion.
si apre il canale, quindi
So
si
the
fa
channel
passare
has
il
been
cloro,
opened,
si
you allow for
ha una
chlorine to pass,
iperpolarizzazione della cellula.
and we have the
Quindi che cosa fa?
hyperpolarization of
Questa
the cell,
iperpolarizzazione
and
quindi
the
va ad
hyperpolarization of
inibire
the cell
in uno
is
stato
going to
di
inhibit
eccitazione,
in
va a
an
inibire
excitatory state
l'eccitazione cellulare.
the
Per cui,
excitation of the
in
cell.
un modo molto semplice, quindi bilancia l'eccitabilità neuronale.
So it is balancing the excitability
Nel
at neuronal level.
35-40% delle sinapsi del sistema nervoso centrale, quindi sono GABAergiche,
So
e
most of the synapses are GABAergic,
l'espressione del recettore
and
GABA
the functional
è
expression
critico
of the
nel
GABA A
processi
receptor
di
is of critical
epilessia,
importance in
ansietà,
the
disordine e anche di ritardo mentale.
epilepsy, anxiety
Allora,
disorders, and cognitive defects.
questa in questo slide, giusto vi voglio far vedere tutte le mutazioni che sono state trovate
Here I want to show you the different mutations found on the GABA A
qui
receptor,
potete vedere anche quelle che sono state trovate nella subunità alfa 1. Allora,
and the one that were found
qua
in the subunit alpha 1.
in questa diapositiva, in un modo
Now
molto
here, in this
riassuntivo,
slide,
semplice,
in a
io ho
high
riportato quello che c'è
summarized
in
way, I
letteratura
was reporting what I found in the literature
subunità alfa 1 del
as
recettore
far
GABA.
as the subunit of
La cosa
alpha 1
interessante
of the
è
GABA
che la
receptor.
subunità alfa 1
The interesting thing is that the
è
alpha 1 subunit
il target di tantissimi farmaci
seems to be the target of so many
ehm,
drugs,
che vanno anche, tra cui anche le benzodiazepine. E
and
poi
amongst
nel
them we
sistema
see
nervoso centrale è
benzodiazepines,
stato visto
and
che
that
in
central
topi
nervous system
knockout
we've
per
seen
alfa
that
1
in the
quindi
knockouts
si ha un maggiore
mice
effetto,
for
diciamo,
alpha 1 we have
si hanno delle
higher
maggiori
effect,
crisi epilettiche.
and
E
we have
andando a
more
modulare l'espressione
seizures
della
in the
subunità
day.
alfa 1 si è visto
So
che
if we
si ha
modulate
una
the expression of
riduzione
alpha
delle
1,
crisi
we've
epilettiche.
noticed that there is a
In altre
reduced
parole,
number
in parole
of
molto
seizures.
semplici, se si ha un aumento in knockdown, cioè nel knockout di
And
modelli
if
animali
with the
in cui non si
knockout
ha alfa 1,
animal
si
model,
hanno
which is not the alpha 1
epilessie,
model,
si hanno
we
crisi
have
epilettiche.
epilepsies, we
Se
have
si aumenta,
epileptic
quindi, se
seizures,
si
but if
aumenta
we
l'espressione
then
di alfa 1,
increase
quindi
the
si ha
alpha
una
1
riduzione
expression,
dell'epilessia,
then
quindi
we
c'è un
have a
effetto
reduction of
inibitorio.
epileptic
Okay,
seizures,
questo
so
in
there is an
modelli animali.
inhibitory
Purtroppo il
effect.
lavoro
This is only
dentro
in
con la
the
pinza
animal model,
è uno, io ve l'ho riportato.
and this is the only work that
Per
we
cui
could find.
noi siamo stati, direi, molto contenti di aver trovato come
So we were
interatore
truly
che la
happy to find
Protocadherin-19 fosse associata
Protocadherin-19 and to see that it was associated
alla
to the
subunità alfa 1 del recettore
subunit
GABA.
alpha 1
Però,
of the GABA
siamo andati
receptor.
poi a valutare e quindi a investigare in più dettagli questa
Then we
associazione.
tended to
Per prima
investigate
cosa
in detail this
abbiamo
type
fatto
of
un
association.
esperimento, se volete, banale. Abbiamo
So we conducted a very simple experiment. We
sistemi non in neuroni, ma in cellule
not at
non
neuronal
neuronali,
level,
che
but
sono
in
le
non-neuronal
HEC, abbiamo
cells, that is the HEC
sovraespresso
cells.
in
We
un esperimento la
overexpressed
protocaderina
the
insieme alla
protocadherin
subunità
together
alfa
with
1
its
e in un
alpha
altro
1 subunit.
set di esperimenti
And in a
abbiamo
different
sovraespresso la
experimental
protocaderina
set, we
insieme
overexpressed the
ad
protocadherin
alfa 1
together
più altre
with
due
alpha
subunità del
1
recettore.
plus other two subunits
Allora
of the GABA
potete
receptor.
vedere in questa, nel merge, cioè che con i colori, diciamo, i
So here you can
puntini,
see that we've
gialli
marked
indicano
with
che
some
le due
colors,
proteine
yellow colors,
sono nello
and
stesso
this is
sito,
indicative of the
ok?
fact
Quindi
that the
sono
proteins are to
associati.
be found in the same site.
Ancora di più lo potete apprezzare quando insieme alla
And you could
protocaderina
even
e all'alfa
appreciate
1 sono
that
state sovraespresse
together with
la
the
beta 2
protocadherin
e
and
la
alpha 1,
gamma 2
and we see the expression of alpha 1
subunità
and
del
gamma
recettore.
2 subunits of the GABA
Dopodiché
receptors.
abbiamo riconfermato questa associazione biochimicamente
then we reconfirmed the associations from a specific point of view. We did conduct some experiments, and this is what we noticed.
Troppo veloce? No, sono andata... Cioè, no. Ecco, ok. Allora, abbiamo fatto anche, abbiamo riconfermato questa associazione biochimicamente sempre...
So this is the association, the biochemical association at the level of cells which are different from neurons, and we did notice that this is the immune precipitation test, which is the test utilized to see whether there is an association between the two proteins. This is protocadherin. This is the subunit alpha, and that they find themselves in the same place. And if the other subunits are overexpressed, then you will find them associated. At the neuron level, we tried to see whether in normal conditions the two proteins can be found one next to the other, and whether they can be found together. Now this is a branch of a neuron, this is a dendrite, and we see the spots, and this is the magnification. You can see yellow colored spots, and this is indicative of the fact that the protocadherin is associated together with the subunit alpha 1 of the GABA receptor. And at neuronal level, with the co-immune precipitation, we've seen that in terms of neurons, and also if we tend to do the lysis of the rat brain, the two proteins are expressed together. And here, at this point, we've seen so far that Protocadherin-19 interacts with the subunit alpha 1 of the GABA. We've seen that the two proteins are together and find themselves in the same site as the neurons. And then the second aspect is that we could evaluate if the protocadherin could be modulating the expression of the GABA receptor. How can this be done? In a very simple manner, by just reducing and blocking the expression of protocadherin by using the technique of silencing, and we associate it to a specific sequence whereby we tend to overexpress the specific turns for the protocadherin. So we did eliminate the expression of protocadherin, and in another experiment instead we did the opposite, we overexpressed it. So we have in one case the elimination of protocadherin, and in the other we have the overexpression of the same protein. And then we tended to look at the levels of expression in both conditions, the level of expression of the subunit alpha 1 of the GABA receptor. Now here you can see that this is the control, because we did control having eliminated protocadherin and having sort of abated it. And here, this is at neuronal level. We see no expression of protocadherin. This is the basal condition of a protocadherin neuron. So if we eliminate it, this is what will not be visible. Now, if you check the two conditions, once we have eliminated protocadherin and we abate it, we see that if we check for the subunit alpha 1, and we see that if we abate protocadherin, we get less of it. We've been using so far two different types of tissues. We use the cortex, so neurons taken from the cortex, and neurons from the hippocampus. The two are the regions where the learning and the memory processes are being performed. And that means that we have abated the overexpressed protein in younger neurons and mature neurons, where the synapses tend to be active, functioning, and complete. After that, we checked the two conditions, and we looked at the alpha 1 subunit of the GABA receptor. And we did see that the GABA receptor comes with the same expression pattern, same as the one in the protocadherin. So if we abate protocadherin, we have less alpha 1 from GABA. If there is an overexpression, we have more alpha 1. And that's in two different conditions, in both tissues. So here we have the expression pattern of alpha-1 in normal conditions, or this is what you get if you eliminate protocadherin. And you can see that in this case it's reduced. So we see that from the biochemical point of view, from the staining point of view, from the morphology point of view, it is easy to check. And the other thing that was also checked was the level of expression of GABA at the surface level. So the GABA receptor is functioning and is active if it's located at the surface. It will also perform as an ion channel, and the role of the GABA receptor during the synaptic transmission is not accepted. So after that, we wanted to check whether the level of the protein, what the level of the protein expressed was at the surface. And we saw that we have less alpha-1 receptor at superficial level. So what do we think? We are trying to understand how and what Protocadherin modulates at the GABA receptor level. So what is the molecular mechanism behind this modulatory effect? This is associated, certainly, but this is regulating the transportation of the GABA receptor within the membrane. And here I want you to see what the GABA receptor is. We know that the scheme is highly complex, but the hypothesis is that between the receptor and the protocadherin, through some cellular mechanisms, and we expect the KIF5 to step into the place. So protocadherin regulates the transportation within the membrane, and this is the delivery of the membrane receptor. So our study is targeted to understanding how the protocadherin may regulate the traffic of the GABA receptors. And then, at the same time, we want to see, because our data until now were in vitro data, that is where the neurons are being cultured. And obviously we want to study them in vivo, that is with the animal. We want to see the modulatory effects of protocadherin at the level of the GABA receptor. And our approach is that we have been currently using two different approaches. One is the one whereby we block the protocadherin expression in the acute situation, i.e. by using the electroporation in uterus within the womb, and then the other one is that we will be silencing the Protocadherin-19 in adult animals by using the stereotactic injection, whereby we tend to abate the protein locally by using the stereotactic technique. And with these experiments, we are going to see the effects of the block of the protocadherin at the level of the development. And here instead we tend to check for the acute effect. If we look at the experiments that were done until now, we see that they were obtained by just simply blocking the expression of protocadherin by using the electroporation in the womb, and in pregnant rats. We tend to adopt specific electrodes. We silence given areas, and we also silence the expression of Protocadherin-19. And we then see what this entails from the morphological and functional point of view. We can see quite clearly that there is a given effect. So we will have to see how, in normal condition, we have a given development, and then what happens if we do silence the protocadherin. So here we have the situations. This is the image of the hippocampus. In green, we've shown the imaging of the neurons, and with the silencing of the protocadherin, this is what happens. You can immediately appreciate what happens here. There is, as a matter of fact, a non-appropriate localization of neurons. Most probably there is a reduction in number of neurons, a non-appropriate localization, and a defect of the migration. So this is what we observed at cortex level as well. And with the in vivo experiments, what we intend to do is to try and see from the functional point of view what this entails. So here we will see some different experiments, that have already been put in an agenda. We want to see whether they allow us to measure the currents of inhibitory synapses and excitatory synapses. We want to see whether it is possible to have a rescue, i.e. bring back the conditions back to normal by administering specific agonists. And here we can only imagine that there is a defect at the inhibitory GABA currents. And if there is a defect, then we would administer an agonist of the GABA receptor to allow it to go back to normal, to its normal functioning. And together with this, we need to have some behavioral studies conducted. And at the same time, we also want to see if in the adult, and this is the stereotactic image that was given to us by the association. We wanted to see whether if by just simply silencing some specific hippocampal areas, so if we administer this type of expression in the adult, we wanted to try and see if we have an acute immediate effect. So we wanted to block in the rat of the protocadherin expression, and then after a week, we saw whether it was possible to sacrifice the animal and then do a specific study as to what happens to the protocadherin. So this is a functional study, which is also looking and checking onto the behavioral aspect. After that, we should try and establish exactly our conclusions. So we here have some preliminary data, but then if we go back to the initial panel, where we put the emphasis on the imbalancing of the synapses, and we see that this is the situation we find ourselves in, because somehow the defect, the functional defect of protocadherin is then associated to a defect of the expression of the GABA receptor. So this is a defect which provokes a reduced GABA receptor in the surface. We do not have the balancing effect in between the excitatory ones and the inhibitory one. And we've seen that there is a state of higher excitability. Now, obviously, here things have been oversimplified. This is just one of the many mechanisms. And we've found the alpha-1 receptor of the GABA receptor. And this is what then generates a different idea. And this is having to do with the possible mechanism of action of Protocadherin-19. Let me conclude by saying that the person who leads the project is Silvia Bazzani. She was given a fund from the Accademia dei Lincei, and she's now also been supported by Teleton, and she is the one in charge of the project. Apart from that, I would also want to thank the foundation for this splendid gift, and hopefully we will start soon making experiments. Thank you. Many thanks, Dr. Passafaro. Do we have any questions for this presentation? Seems it was really interesting. So let's see if there are any questions from the audience.
So I was wondering if you think that Protocadherin-19, one of its roles is to stabilize the GABA A receptor, how you think that might work in boys? Because they have the mutation, they don't have any Protocadherin-19, but they're largely unaffected.
Yeah,
And
this is a big question. Maybe there is some compensation for some other. Yeah, this is a big problem. I agree with you because there is, but one possibility is there is some compensation from some other. Who knows? This is, but maybe we can discuss. Joseph.
Thank you, Maria. This is lovely work.
Oh, no, it's very preliminary results.
So the question is here, where we discussed it very quickly and briefly, whether PCDH19 can interact with other GABA subunits. And the other question is more clarifying. Is the GABA alpha, the alpha-1 is only synaptic or is also extrasynaptic?
It's also extrasynaptic.
So the interaction you have seen, the localization, was it a synapse or extrasynapse?
No, actually with this resolution, you cannot distinguish very well because they are very close to each other. But there is some partially synaptic and maybe there is also extra. But the idea is to look at first the GABAergic currents, but also the tonic currents, because this is yes. This is the functional experiment we need to show some, but we can discuss and to do some.
Thank you for this very beautiful work. I would like to know whether have you the same distribution when you co-transfect the gamma-2 and beta-2 subunit?
They're actually in heterologous system. Okay, they are all together, but they are together. Yes, they colocalize very well. Now the idea that actually we want to do now the electronic microscopy and to see in high resolution if all the complex are together.
Because
this is the only. Exactly, with the electronic microscopy, we can clarify very well at this point.
Because the gamma-2 subunit is very important for the trafficking and the localization of GABA A receptor.
Yeah, I know.
Thank you.
But this resolution seems to be...
I have a curiosity. I'm curious about something. How could you end up thinking of the GABA, and how is it that you chose the alpha-1 subunit? And then, is it possible to conduct electrophysiological studies? No, the electrophysiological studies are now being conducted.
Gli studi di elettrofisiologia
and that's the functional studies, in vitro and in vivo, have not been highlighted, but Silvia will already be programmed to do it. And as far as this identification of the alpha-1 subunit, I can tell you that we did use the screening in order to localize it. I was not trying to annoy families, so I didn't point at it. But here, the C-terminal of the subunit is what will then allows us to have the human brain library, and then we will have to look at the number of territories. A tiny technical question now, just to make sure of things. When you consult a library like this, do you also do a ranking, which is a quantitative one, or not? Now, we know that there are new libraries, and we tend to check the interaction of the proteins within the yeast, which is immediately visible. But then what is to be seen is the screening of the interactors. This is something that is mandatory, that should always be done. And then maybe we could choose a method which is a proteotic interactive method. And then otherwise one would run the risk of finding a potential quantity of interactor and not have a ranking. Now, this is a system which allows you to
degli interattori. È chiaro, se ne possono trovare 20-30, dopodiché bisogna fare
To
la
find some 20, 30 interactors, then you have to differentiate them, and then you have to check by conducting immune precipitation experiments. And the proteomic is something which entails an experiment where we can find hundreds of proteins, but this is not really meaning that all of them are interactors. They may be sometimes only associated. So if we instead conduct the interaction experiments, then we will end up with a precise and accurate answer.
Ok,
Bene.
andiamo avanti. Prego, una breve domanda.
A very quick question. I must admit how ignorant I am with this respect. And you've been showing this relationship in between the PCDH19 and the gamma alpha 1. Now, the reason why we tend to go through one study to the other is due to the fact that there seems to be no possibility of verifying the same relationship with humans and patients in general. As far as humans are concerned, let's say that the science seems to be proceeding by steps. So from cells we switch to the animal model. After that, if the results you've obtained until then are passable, then maybe we could have some PS as other people do. Nonetheless, this is directly human, and it is only possible to see this with the cultivated cells, PS cells. And this is the alternative, because within this context, the patient has no alternatives. What if we take a specimen? We still have to have PS, then differentiate them. We should then differentiate the PS cells, and then we should check what happens at the GABA levels, the GABA A levels. And this again is something that can be done. Or as Joseph does, he is immediately prescribing the drug to the patients.